calling, and there are two subfolders. There are two data sets that you could practice calling. We're going to do the 301. So we're going to open the 30 WES variant calling. And the notebook we're going to be opening is a variant calling JATK. But before that, just show you that three folders. So each one of them is a different sample. Inside of that folder, there's an aligned folder. Inside of an aligned folder, that is the FASTQ files. I also have the tendency to make them more or less independent. So if one day you come back, I'm explaining the same thing in several Jupyter notebooks. You've seen these before. So I don't think I need to explain how. Oh, yes, I need to explain again. It says this is going to be running commands in the terminal. This is not R. Okay. We are using the Python 3 kernel, and this thing here is to tell that we are running a command line, a bash command. You could do all this, I think I said a lot of times this, opening a terminal and running the commands in the terminal. It's the same thing. There's no difference. And even some people tells me I shouldn't do this. I should teach you how to use a terminal instead of using a Jupyter notebook. I don't know. I like the notebooks because you have the text. So all this is just to show you how a variable works in Bash and how you assign them, and how you use them. We've seen this before. The idea of how we run commands. Generally, we go to the folder where the software is. You run that software. We don't need to do that. Because everything is in the path variable, right? You all remember this, right? If not, tell me no. How to get help. Yeah, here is the idea of the path. And why in Conda we don't have to. So I like to have it that in even if we just mentioned this not long ago. In case you go back to a single notebook and then you don't remember why we're doing things, you always have it on top. Oh. It's only me who lost connection to the Wi-Fi?
It's me too.
Okay. Well, more or less, you see, I'm just listing the steps of the analysis. And I also put links in the notebooks how to get more information too. So here you have the JTK best practices. There's a lot of documentation. There's a lot of FAQs or people making comments, and there's a lot of answers for common problems too. Is it back? I don't know. It's not? Okay, sure. No, not a problem. I'm just asking. So these are the steps. These are the steps that we're going to do today. I explained them in the slides, but here we have them in the right order. So we're going to do a FastQC. We're going to align them. We're going to mark duplicates, and that's something that I didn't say in the slides. We don't remove the duplicates. You could remove them. We just mark them and say, this is a duplicate. Don't use it. Base quality scores recalibrations. We're going to do variant calling with this tool that is called haplotype caller. All this is JTK, by the way, but they have tools. JTK is the tool set. We're using one tool inside of the tool set that is called haplotype caller. We're going to join genotype all three samples to have a single file. We're going to have some steps of filtering to remove variants that we consider probably are not real. And we're going to annotate them with BEP, variant effect predictor from ensemble. Oh, it works now? I don't know if I need to restart now that I'm thinking, but. So everybody's back to the notebook. I think I mentioned this, but just in case, because you're going to see them a lot, I think, in this notebook. The command line expects your codes in a single line. Normally, there's even exception for that. But a way to break the line is using the // symbol. And it assumes, it says, wait, don't execute it. Let me keep writing. Wait, not execute it. Let me keep writing. Now you can execute the line. It's a way to break the line and make them more readable. You're going to see this often I think you see this often in this notebook. And what we said about the samples, another thing that we don't teach enough maybe is how to be organized in genomics, because you're going to get a lot of data, and it's hard to be organized. I know some people come from different fields, and I assume things that people know, and I'm realizing that it's not the case. And we create a lot of folders with long names, and then subfolders, and it's good to be organized, so you need to be organized. Do we need to do this one? No, right? We've done it a lot of times. You know how a FASTQ file looks like. Okay. Because you know how to run FastQC. Random for, you don't pick at least one sample or random for one sample. And if you don't remember how to run the command, you can figure it out. You have ways to figure it out. Is the text readable in the back? Should I go up a little bit? Problems running FastQC? I won't be here next week, so don't expect me to tell you how to do everything. We say that, right? Teach how to fish. Don't give the fish directly.
Here we can give the name, the above name, father, mother. We can reuse that file.
You need to provide what? No, no, no. Just run it. It would automatically produce the output where the FASTQ file is. That's fine.
So like in the above they have mentioned father file. So we can give the name.
You need to provide to run FASTQ. You need to FASTQC. You need to provide the
File.
FASTQ file.
File, yes.
Yes.
So there is above in the another before this step there is one file.
There's no BAM files yet.
But father are one file. Father.
You can do whatever you father, mother, provan. Just do at least one.
Okay.
I know you did the homework, so I don't need to tell you how to navigate folders. It's the last day. I assume you have 5 days to do homework. It's fine if you just do one. You done it? I'm so proud of you. If one thing you're gonna do right, at least at the end of this, is run FASTQ. At least that's good. Father lowercase. Does it have autocomplete?
We have mentioned father, mother. You have to give the name of the student, right?
Remember that the trick with the tap. You can do autocomplete. You don't have to type everything. I'm assuming all of you were able to run FastQC, right? Good. So FastQC, you need to provide the path to the sample. And because all of you know did the homework, I don't need to explain you how folders, subfolders work. Bonnie is looking at me like, oh, you're too harsh. So if you run it, you're gonna. So what is the challenge that you had running this? Tell me. What has been the main challenge? Why you, if you got stuck, what was the reason that you got stuck and you weren't able to run it? Because this is one of the simplest tasks we're going to do today. What was the problem? I don't know, somebody had problems, right, running this, I'm assuming? No? So we provide the name of the program we are using, and the only argument that we're giving to the program is a FASTQ file. So we need to provide the location where that FASTQ file is, which is in the father folder, or mother program, whatever you choose, and a line, and then the name of that file. And that's what you see on the screen. And if you run it, you obtain the HTML report that we've seen 100 times. Anything that called your attention? So these samples, I forgot where they're coming from.
So mine is we can save it, right, as save PDF, or it is automatically
Yeah, automatically generates a report in the folder where
path is there.
the files are.
Yes. Now this is the file.
Anything you want to comment on? What do you think? They look good? They look bad? They're more or less good, yes. There's something weird though. Yeah. I don't know the reason. I need to tell you, by the way, this is whole exome sequencing, but we are not aligning the whole set. We are aligning only one chromosome. Data for one chromosome to simplify the process.
Yes. So in this, the score is approximately 20. This Z score. So this
You
is
mean
20.
40? You mean around 40?
So score is almost near about 30. So this is good enough for the
So when you say score, you're giving only one number. Think about this. This is all the sequences are here, and there is it's a box plot or histogram on the verticals. So the average and median must be close to 40, right, or not?
Close to there is a 35,
30,
35.
Okay,
35.
it is good enough. I mean, sequence
So we can
is what we have. We don't have a choice today to
in
resequence or anything like that.
the transcriptomics we usually do the trimming for these
Yes,
lower scores.
but we are not
Here
going to
we
do
are
it.
not doing.
No.
So there is no adapter content, so it needs to be.
Of remaining sequences if reduplication is removed.
We are not going to remove them. We're going to just mark them, but it's the same thing at the end. So your question is if there's going to be enough left.
No, how many should be left? Like 70,
Oh,
80%?
how much is the percentage of PCR duplication? I don't know in this case, but it's a good point. You always need to check that, right? So if your PCR duplication is too high, you may have a problem. It may be that the sample is very amounts, or you need to change something in the protocol, or there's a problem with the sample. So PCR duplication, it's important to check, definitely. The levels we're talking here, the
So
percentage.
like if there are not many duplicates, it's fine.
Yeah, I
95%.
mean, sometimes you have to do what you have to do, right? So questions? Should we move on or not? I don't know. I have the feeling nobody's telling me anything. I don't get feedback. Need feedback.
So can you explain this? Here
Yes,
per tag sequence quality.
sure. Um, I'm getting a question to review some of these plots. Any questions about the first one? No, we've seen it before. It's telling us about the quality of all sequences per base. We generally want it on the green area. That's what FastQC telling us. It doesn't need to be there necessarily, but and you know that Illumina should go as high as 40. I'm not sure why we see 42 here. Again, I obtained this data from somewhere, so I don't know how it was sequenced. The question was per tile sequence quality. This is the location where the sequences are coming from. If, for whatever reason, so coming from in the flow cell, if for whatever reason some area is bright, like concentrated bad sequencing in an area, it's going to be flag, telling you, oh, there's a problematic section in the flow cell, because we know the location. Yes? So it should be, ideally it should be just blue, right? There's nothing here to look at it. There's always something, but. So it seems that, I guess, the problem is around here. That area of the flow cell produce a bunch of bad reads, or low quality reads. So next step. Yes. Yes.
Then also we should skip the trimming part or
So a good point too here. I mentioned not to trim by quality, but why we don't trim by adapter contamination. Again, look at JTK documentation. They offer tools for some trimming. We don't. We assume that the BWA is going to soft clip the adapter. But it's true that the first bases of the adapter could also align to the genome and produce a call or something, right? Yeah, we don't trim. Sorry, what is the question?
not use stringent parameters.
No, I mean, again, check. I would recommend that I think JTK, they have other tools, or I remember reading about it.
Because we used to do trimming always.
Yeah. And for JTK, I wouldn't recommend it for that, because you're modifying the read. You're not modifying the quality scores per se, but you're modifying the read. And it's good to have all the information that you have for all sides in that region. And I'm not going to call an expert either. So, what I do, I work in so many different things that I'm not going to call an expert of anything at the end of the day. I don't know what I am an expert of. So we did a quick fast QC. In any case, because we are not going to trim and we don't have a good reason to believe they're terrible, we're going to have to work with these samples. We don't have anything else. We need to report our case to the clinician and tell him, oh, this is the disease-causing gene. So there's not much we can do now. So next step is going to be alignment. There's going to be a lot of text to read if you want to, and I recommend you do. For alignment, we're going to use BWA MEM, and we're going to use pipes again. The pipe symbol, the vertical character. So remember, we take the output of one thing and we give it to the next software to do something with it, and we give it to the next software with it. And we have three pieces, right? We have 1, 2, and the third one here at the end. Probably the ugliest and the one that you think, why we're doing all this, is this one here. This is needed for GATK. And I tell you all the things, what it means in the text, so I recommend you read it. But the main idea is you need to label the sample that it is, what platform produced it, and so on. You need to provide some information for GATK, because it uses this.
The text they have mentioned, the add to uses 2 threads for sorting.
Yes, I ended up modifying that for alignment. Yes.
2 threads for sorting means?
Did I set sorting? Oh, I did. I didn't modify this. Let's see what happens. We run it. Initially, I decided that we would use thread. is a way to parallelize the analysis. Thread meaning, imagine you have two CPUs, you can use both of them. There's software, not all software can be parallelized. In this case, we know they can be, both of them. So you can indicate how many CPUs you want to use. That's what we're doing. Both some tools and BWA can be parallelized. If you have really enough compute power and you want to parallelize at big levels, you can indicate as many CPUs as you want, and it's going to go faster. That's the main idea. Dividing the job makes the thing go faster. Is that the question? I'm answering your question?
So here we are using two types of threads.
No. The thread means you are dividing the, I'm going to simplify it in the way that you are dividing the jobs into two sub-jobs to run them in parallel. That's a thread mean.
So we have run these files, unaligned files. Now this SAM sort will give you the output file. The output is mentioned as a U, right? Output
Read it.
unaddressed RAM. So it's not this. O is OBAM. Okay, output file is O. OBAM.
So another important thing, if you have to repeat this in your computer, read the arguments, because the ones I'm using here is for simplicity sometimes and for the setting that we're working with. If you have more compute power, you want to parallelize even more if you can, right? So do it. You can probably work with compressed BAMs if you want to. There's a few things that you should test yourself and see what it works for your setting. What I'm indicating here is what we can call maybe the best practices steps for analysis, but the arguments, you need to adapt them to your situation. Like memory, I think this should be fine in any setting, but you can see if you get an improvement if you change it. So what we're doing here, any questions about any of the parameters that are answered up there? But so all the arguments should be explained. Didn't I explain that one? Yeah, I did. Oh, sorry, I keep doing. So here. So, well, again, so. So, oh, again. Soft clipping is when the aligner is not able to align it, and then we are saying you need to consider that in the other aligner, in the other read. The complementary would be the other. So we have two reads. They map. You're soft clipping something in one, you need to consider soft clipping in the other one too.
Is there a reason we're sorting the output?
Yes. Let me run it. Is everybody able to run it?
Yeah, but it's taking a
Just
while.
one question here with combined HS, like earlier you have explained that like as a single.
Why we do it in a single command and not separate? Is that the question? And I'll answer yours too.
Like HUS as combined, or it is like supposed to be put together, or like as a separate.
You could run it separately if you want to, but it actually is a little bit more complicated if you run every step separately here. So we're doing three things. First one, alignment, and it returns a sum. Nobody likes to work with sum because it's too big. You could transform it to a BAM file and compress it. That would be this, or not compress it in this case, but you could. And then we need to sort it. The question is why we sort. For quick access, and a lot of softwares that we're going to use downstream, they want the BAM file to be sorted. and indexes. Did it finish for anybody? It should take 5 minutes, I think. Still running? And how long did it say? Because if you want to know how long it takes, oh, it doesn't give you something readable? I thought it gives you something readable.
Oh, wait, okay. 586 seconds. 586.
508. That's not 10 minutes, it's less, right? So it's 8 something. It's going to take 9 minutes, more or less. Yep.
So we can do these multiple steps.
Yep. Separate.
We can also separately do this.
Yes. But I don't think it's more work to do them separately, if I tell you. Because you're writing intermediate files and you're going to may need some view. At least you're going to combine 2 of them, I would say. But you can do all 3 together.
Simply agree, but we need to do more work.
I think it's more work if you try to do them separately. I know you prefer me to explain things. Sometimes I'm going to take a rest from now on, or every now and then, and I'll let you read. You can, even if it's running, you can prepare the code for the exercise that you just have below. Oh, you're running all 3? Then you can do. Oh, it's done? Yeah, then keep working with the next exercise. So we only aligned the first one. We need to align mother and proban. So go ahead and align them. Even if it's running, if your code is running, you can start typing the align process for the other 2, or copy and pasting, right? Don't lie to me.
I tried to copy it, it's all gone. So do I have to type it again? I mean, like I was trying to copy,
Yes.
and the whole command is gone over here.
Oh, you can do Control Z to go backwards, and it should come back. So if you go to the cell where by mistake you probably remove it in Control Z.
Command Z, I think.
Yeah, Command Z.
Those what?
You need to be in the cell.
और एक मतलब 4-5 बस जब तक वो आता है तब तक कर लो और 10 बार कर लो
Okay, I'll put it back on the screen so you can see it. Um, I'm surprised that the Control C doesn't work, because if you type something, if I type something, I can do Control Z and remove it. I'd send you an email with the code.
I sent it
Okay.
already.
So it seems that it takes for some people in between 9 and 10 minutes. So again, work the code for the next step because it's going to take a long time. That's what we could do. Probably we could finish this section, let it run, and then we go for lunch.
You have mentioned doing for other samples. Do we need to open a separate notebook and do it, or we cannot do it in the same file? We can.
About the other folder.
Yeah, the other folder.
I mean, you could work in the same document. It's not going to look
It's
clean.
confusion. Yeah.
So you could copy the document and put it in the other.
Okay.
The other thing you need, the other folder only contains one sample. So you have to remove everything related to multiple samples.
Yes. Yes.
If we have time, we can work in the other document too.
Next. Next cell. उसी फोल्डर में जाएगा ना, मैप उसी फोल्डर में जाएगा। हमारा जब फादर का फोल्डर में फादर ही जाएगा, मदर का फोल्डर में उसकी मैप वहीं जाएगी। आपके पास क्योंकि तुम्हारी वही है। ऐसे ही एक होगा दूसरा चला जाएगा।
Does the server like run every command which you mentioning? Because like I was wondering, like if we have like a terminal open separately, like if we have a terminal open separately, can't
Yes.
we just like run on like one of the files while the Jupyter notebook is running this one?
Yeah, you could.
Yeah, okay, let's do that.
Yeah, but the problem is, don't do it, because there's going to be 40 people trying to run something. Let's do sequential.
Yeah.
I don't want to get that situation we got the other day that we were using 100% of everything, and it seems that it went even slower. I mean, I don't know how much we're using now, but I'm sure we must be 100% too. Oh no. Well, yeah. I don't know. It seems. And we don't see all CPUs. I don't remember how much. No, yeah, we see all the CPU here. All 48. All are busy. You can guess already how to align the other two, right? Be sure to modify this line. And also remember, you can press tab to autocomplete if you don't remember how the files are called. Oh no, that's true. No, we cannot because it's quotes. We cannot use tab for autocomplete because it's between quotes. So you need to double check that file names are correct. Yeah, file names are in lowercase, just in case anyone has doubts.
First letter is capital and second small, lowercase.
Yep. Folder names uppercase, file names lowercase. Yeah.
So let's just
There's no reason for that. But Yeah, here it doesn't matter if it's uppercase, lowercase, it doesn't make a difference. They need to be different values though compared with the other samples. You could have named them however you wanted.
So the protocol is same as for T08 Genome system. T download software.
I see a lot of people in this room. Just to clarify one thing, look what I'm doing here. So the father is done, we know, but because there's a number next to it, is the first step that I run in this pipeline. Mother, that you have the asterisk, it means it's running. I'm good. But I'm also running the proband. It's not running, actually. It will run when this is done. So that's what I'm trying to you to do. So don't wait for this one to finish, to code this and run it. Run them. They will run by order. When this is done, automatically we'll come here and run it. So when you have all three, you can have the break. So I didn't want you two guys to wait for this to finish. I'm taking the break.